So the main point of my presentation of my talk today would be that life and health span are not really controlled by the entropy and time progression, but more so by the effectiveness of repair and regeneration. For example, young and old person can have the same amount of damage. In fact, young people could inflict on themselves more damage than old people, but they have much better efficiency of repair. And by repair, I mean repairing cells and tissues, and also repairing molecules inside of the cells. And so the main question that our group is pursuing is, would improving the efficiency of repair in an older person make them meaningfully younger? In other words, we cannot really fight or combat successfully entropy, we are going to lose, but can we, for prolonged periods of time, for many more decades, withstand the entropy by improving the repair of our body. And my favorite example of that, there are animals, for example, rats and squirrels, which have very similar body size and similar metabolism and similar diet, and they look very similar. But squirrels live 20 or more years and remain very healthy and young when they are 3 years old. In contrast, rats live only 3 years, and so they are full of disease and old and unhealthy at the same chronological age. So in my opinion, once we have an opportunity to study squirrels a little bit better, we will realize that this happens because they have much better efficiency of repair. So they're repairing damage inflicted on them, and that's how they live longer and healthier. The next point of introduction is that we are a mosaic of cells of different biological ages. So when we look at ourselves in the mirror, we think that, yes, this is a particular biological age of my entire organism, but if you look with more precision, we will notice that some cells in our bodies are stem cells, and they are tissue stem cells. They are not the stem cells like embryonic or pluripotent cells. They are dedicated to making particular tissues, but they are relatively younger, and they can activate telomerase where needed and expand telomeres of their progeny called progenitor cells. And then there are other cells which are differentiated cells. So those are the cells which experience the most damage because they function and make us alive. So they actually perform all of the things that we consider is important for us. For example, muscle cells are contractile, so they allow us to move voluntarily, and brain cells allow us to think about how we move and other things. So differentiated cells, they experience damage, and telomerase is inactive in those cells. And for all practical purposes, they are the functional cells of our body. And then there are senescent cells represented by this part of the tree stump. And these cells are the most damaged of all. So they have DNA damage, telomerase is inactive, and they express the SAS proteins and secrete them, and they are also resistant to cell death or apoptosis. So when you look at this tree stump, it metaphorically explains what kind of cells do we have in our bodies. What is very interesting is that extrinsic cues or different systemic milieu or different ages of blood can quickly and robustly both rejuvenate and age all of these types of cells. So while we think that it simply could happen from the progression of time, it is not necessarily so, and biological age could be reversed for all types of cells, as I will tell you today. Not just for the stem cells, but also for senescent cells and differentiated cells. And also, very importantly, that can happen for derivatives of all three germ layers, not just for one tissue, but as tested back in the early 2000s by Mike Conboy and myself in Tom Rando laboratory in liver, muscle, and brain, and then as confirmed in many laboratories, including Tony Weiss-Corey lab, Amy Wager's lab, for many other tissues and for these tissues again. So these discoveries of reversibility of biological age come from two main experimental approaches. One is heterochronic parabiosis setting, which is illustrated here by this hilarious slide taken by Eric Worden from Bach Research Institute. and this is Halloween costume. And another is heterochronic apheresis or heterochronic blood exchange. In heterochronic parabiosis, animals are sutured through their skin and develop common vasculature capillaries in about 7 days, and then they remain sutured for many weeks, and then their responses are analyzed. In heterochronic apheresis, in contrast, there is exchange of only blood, and that takes, if you are experienced, about half an hour. And then animals go their separate ways, so we're only analyzing the blood. And that's very, very important because sometimes people think that heterochronic parabiosis also is just blood exchange, which it is not. Because there is also contribution of young organs, for example, heart and lungs, to simple things like oxygenation of the blood in an old animal. And young kidney and liver remove the toxins. Young thymus and bone marrow provide better niches for blood cells. So it is really effects of organs and not just the blood. In addition, typical old mouse usually sits in the corner of the cage and shivers, but when physically connected to the young animal, it has to run around and exercise and also have better diet because it regularly goes to its food and water. So there are many, many environmental and organ sharing changes that take place in parabiosis, and those are separate from the effects of just blood. And those are not present in the better controlled blood apheresis experiments. Additionally, blood apheresis in small animals allows to perform interesting studies because we don't have to exchange blood just between young and old mouse. We can exchange blood with any designer fluid, shown here as the tube of designer fluid. And this is the paper in press right now in Nature Protocols. So these are introduction on the experimental technique. So the next question is, what is important? Young blood, old blood, both of them. And in relevance to my talk, you can look at it as half young, half old glass of blood metaphor, because whenever we perform parabiotic studies or blood exchange studies, that's what we end up with. The glass which is half empty, half full, but instead half empty, half full, it is half young, half old blood. Many experiments that were performed before suggested to our group that both in vivo and in vitro in cell culture, the outcomes are such that old blood dominates over young, and it inhibits the health and performance of multiple cells and tissues. And then very recently, a great paper from Professor Irina Conboy came out and her group that did this experiment that we were always asked about in my previous talks. So what happened is that they had mice, young and old, in heterochronic parabiosis. And then they kept them parabiosed for 3 months and then disconnected them and studied their lifespan. In other words, would young blood that was shared by old animals make them live longer? Or would old blood shared by young animals make them live shorter? And as you can see here from these results, which are super important, there is no improvement in the lifespan of old mice that were parabiosed to young animals, not at all. So here they are. But there is significant decline in the lifespan of the young mice that were parabiosed to old mice, as compared to how they live in controlled young to young parabiosis. And again, it's not just parabiosis, they were joined for 3 months and now they're disconnected and the lifespan is measured. So this again suggests that old blood dominates over young, and that's why our group is focusing on the old blood. This paper was published online, and it is coming out in the August issue of Rejuvenation Research. And as a new editor-in-chief of Rejuvenation Research, I am asking you to submit your good work to us. We are striving to improve the impact factor of the papers that we publish. And your help in submitting your manuscripts would be much appreciated. And still, I understand how it is intuitively very easy and tempting to think, as based on this cartoon, highlight of The Economist, of one of our papers actually, how it is tempting to think that you can improve situation for old people by simply adding young blood. But because of this evidence and the evidence shown in my talk, this is not our focus, and we believe that the better focus would be on understanding the deleterious consequences of the old systemic milieu on young cells and tissues. So in this regard, as I mentioned, our blood exchange setup allows us not to exchange young blood and old blood, but to have any sort of designer exchange fluid and test its effect on cells and tissues. And in the first set of experiments, we did very simple analysis by replacing 50% of young or old blood with saline, so basically diluting blood plasma. And we added back 5% albumin, which otherwise would be lost by this procedure. And the blood cells were returned to the animal isochronically. So the blood cells are the same age. And what we published in the last couple of years, in 2020 and 2021, is that all plasma dilution rejuvenates all of the 3 germ layer tissues. So all the effects that we have seen in parabiosis or blood exchanges are repeated, recapitulated, if you simply dilute old plasma. So in mice, we demonstrated rejuvenation of old muscle with less fibrosis. In liver, there was less fibrosis and adiposity. In brain, there was better hippocampal neurogenesis and increased decreased neuroinflammation and brain senescence, and improved cognitive capacity. And we have initial data with just 3 people that demonstrated that there is rejuvenation of blood proteome as well. So this is published, and I'm just summarizing it. This work, particularly for people or therapeutic plasmapheresis, was done through our collaboration with Dr. Dobry Kiprov, who performs this procedure in people and shares blood samples with us. So then, just to show some of the results, for example, from this paper, old plasma dilution reduces brain senescence and improves cognition. We have these settings where we exchange or dilute old blood or young blood with 50% of the saline with albumin and the same cells, or we have isochronic control experiments. And what we test is the short-term memory in novel texture and novel object recognition. Typically, old mice, as compared to young, are not very curious or inquisitive, and they do not even know which one is old, which one is new. But after a single procedure of the neutral blood exchange, neutral age blood exchange, they become virtually, statistically indistinguishable from young animals. And that is the strongest rejuvenation that I have ever seen, because it's not that they're slightly better, they're statistically the same as young animals. And that is just from the dilution of old plasma. We did not introduce any other factors. By the way, this procedure, we termed it neutral blood exchange because the age of what we exchange doesn't have any age, it's neutral. And we also show that we can reduce senescence of the brain. So this is SA beta gal cells in the old brains, old vehicle or old exchanges. And one dilution of old blood plasma reduces senescence of the brain to the same effect as a known senolytic molecule ABT263, which is quite interesting. It is interesting because ABT263 does not cross blood-brain barrier, and that will be important to remember in one of my subsequent slides. Neutral blood exchange also reduces neuroinflammation. So these are all CD68 positive activated microglia cells in all brains, and then their amounts are decreased significantly, once again making old brain statistically the same as young in this parameter of neuroinflammation. And neutral blood exchange also improves hippocampal neurogenesis, or proliferation of the neural precursor cells in this particular region of hippocampus known as tentogeres subgranular zone. And once again, from the very low neurogenesis of the old animals, neutral blood exchange makes neurogenesis statistically the same as in a young animal. And we published the same for muscle and liver. Interestingly, the same senolytic ABT263 does not improve hippocampal neurogenesis. So elimination of senescent cells has different outcomes than old plasma dilution. And none of these treatments cross blood-brain barrier, right? So ABT263 does not cross into the brain, but somehow the numbers of senescent cells are diminished, most likely because there is less of them in the periphery, and so there are less of SAS proteins that go through blood-brain barrier. Blood dilution or plasma dilution does not really cross blood-brain barrier. We simply dilute plasma in the periphery, and we diminish senescent cells, and we also improve neurogenesis and diminish neuroinflammation. So there is clear difference between using senolytics and diluting all plasma. And in a later couple of slides, I will hypothesize what that might be. Before I move to this part of my talk, I would like to give one slide on the blood-brain barrier on chip, since it is super important for our studies and perhaps would be of use to other researchers. And this blood-brain barrier on a chip, BBB on chip, was developed by Kiana Aran from Keck Graduate Institute, who is a professor, associate professor there. She did very brief but spectacular short postdoc in our laboratory, and here she's shown with her another invention on CRISPR diagnostic chip. So what Kiana did, she designed blood-brain barrier on a chip, which has all these various cells, but also allows shear stress to be modulated, and continually measuring how the shear stress and age of the circulating cells will influence the integrity of blood-brain barrier. And the data with mouse erythrocytes and human erythrocytes or red blood cells demonstrate that if you look at the low shear, there is not really difference between young and old cells and their effects on either permeability of the FITC dextran or in TEER, which is transepithelial electrical resistance. So then if you increase physiologic shear, now, first of all, you see that even young erythrocytes make BBB less stable, decrease its integrity, and particularly old shear erythrocytes have deleterious consequences on blood-brain barrier. And you can see it by the permeability through the blood-brain barrier or by the diminished TEER. And the same conclusion could be derived with human young and old erythrocytes. So blood will have bad consequences, or blood cells will have bad consequences on the blood-brain barrier, and that is most visible when we have more physiologic shear in our organ chip. Okay, so with this intermission, we now move to the main part of today's presentation, which is on this interesting question. What is the role of senescence and senescent cells in all of these phenotypes of heterochronic parabiosis and blood exchange? And when we dilute old blood, does it simply mean that we reduce SASP and reduce the load of senescent cells in blood, and that is the only and the main outcome of blood heterochronicity? We are usually asked these questions, both Judy Campisi and myself, and we have been thinking about it for a while, and now we have an opportunity to answer it with experimental data. So this is the very recent paper, just came out a few days ago, and the work has been conducted by Oki Jion, who is a professor of her own laboratory in Korea right now. And she did postdoc with Judy, and before that, I think, if I'm not mistaken, she did studies in another wonderful laboratory of Jennifer LSF. So Oki did the lion's share of work on this paper. And the first data shows that old serum makes young cells senescent. You can see here is the outline of the experiment. We can have serum from mice or people, and they could be young or old. And we expose cells in culture to this serum. And if they're cultured with young mouse serum, they have less of P16, and with old, they have more of P16, and this is very quickly in just 6 days. And the same is true about P21, IL-6, MMP3, and there's a diminished levels of lamin B. And cells cultured with old serum has more senescence SA beta gal, and they have less proliferation based on incorporation of EDU. And this will be true both for the mouse cells and for human cells and serum. So that's quite interesting, right? Because we thought that perhaps senescence relies on the long time or sometime accumulation of cell damage, but here we show that it could be very quickly in 6 days induced in cells which are otherwise young. Even more importantly, old serum dominates over young when they're mixed. And these are the observations that Mike Conboy and myself made back in early 2000s, that old serum mixed with young serum dominates. So young serum doesn't have any say, and old serum then has its negative effects, even when they're mixed. And this is the actual experimental data that came out recently on that. So if you look again at P16 levels, P21, IL-6, matrix metalloproteinase 3, lamin B, by all of these parameters, if you look at the effects of old serum, it's deleterious, deleterious, deleterious. And when they are mixed, young and old, it is similarly deleterious. And the same is true if you look at cell proliferation. In young serum, cells can proliferate well. In old serum, they don't. Young and old, they don't. So that then questions this point that adding young blood or serum to an older individual would be useful. And it is another interesting point that what is that with the old serum that is so negatively dominant? Well, first of all, to confirm the paradigm, we also show that old blood makes cells and tissues of young mice senescent, which is true for muscle, for kidney, and for liver. And these are the measurements. And this is, again, super interesting because we thought that senescence requires progression of time and accumulation of cellular damage, but no, these are young mice, these blue ones. And then this is exactly the same chronological age of mice, the orange young old ones, but senescence is induced in just 2 weeks before there was any time to have telomere attrition or accumulation of DNA damage. So senescence is really a trans phenotype, and it is controlled extrinsically. Another interesting experiment is that if we use senolytic dasatinib Herceptin or ABT to 63, we can dramatically reduce in-trans induction of senescence. So if old mice are treated with senolytics before blood transfer, then senescence is not really induced very much. So senescence clearly begets senescence. However, if you look at many other parameters, for example, gene expression for P16, P21, and other genes which are considered to be progerotic when elevated, senolytics have really kind of wimpy, very weak effect. And that, again, to remind you, is not senolytics applied to old mice, but senolytics that are applied to old mice before their blood is transferred into the young mice, and to measure the induction, in-trans induction of aging. So senolytics have very powerful effects on the in-trans induction of senescence in young mice. They do, but much lesser effect on the in-trans induction of aging phenotypes. The same is true if you look at muscle. So this is gastroc, TA, and 2 types of senolytics that we use that act by different mechanisms. Once again, if you look at these orange bars, in some cases, they are much less than blue. In other cases, they are similar to blue. So the overall conclusion is that there are some things which related to senescence and aging. There are many other things that neutral blood exchange takes care of that are unrelated to SAS for senescent cells, that senolytics cannot get rid of those. But once again, senescence begets senescence because now if you look just at the amounts of senescent satellite cells, and old animals are treated with senolytic before blood transfer to young animals, senescence is dramatically reduced, as is visually shown in the upper panels, where this is, for example, satellite or stem senescent cell based on its location. And then after animals are treated with senolytics before blood transfer, their numbers diminish. And of course, the numbers are induced in young animals transferred with old blood. Additional cell types in muscle that become senescent or are senescent are the inflammatory CD45 positive cells, which are most likely macrophages, and we see it here by CD45 staining. So couple of summaries here is that, first of all, we see senescent muscle stem cells, which is great. It is important to know that they exist. Secondly, they are induced in just 2 weeks after young mice have been transferred with old blood once. So once again, there was no long-term damage. It is just old blood inducing senescent cells. And thirdly, if old mice are treated with senolytics before blood transfer, their numbers decline. So very, very clearly, senescence begets senescence and trans very quickly. And with respect to physical endurance of young mice transferred with old blood declines, which could be seen by the treadmill, involuntary treadmill exercise, less so in their hanging test. And once again, treating old animals with senolytics before blood transfer only mildly ameliorates this decline. An overall conclusion remains that when we dilute old plasma, we are not simply reducing cells. We are doing something else, which is quite interesting. In confirmation of that phenotype or conclusion, I told you that dilution of old plasma, and that has been published, make old muscle statistically the same as young, and the same is true for old liver and old brain. And that cannot be said about the effects of senolytics. So this is the schematic of the experiments that Otki did, which is treatment of mice with senolytics, and then some interval of time, and then the mice were injured in their muscle with cardiotoxin, and then we analyzed the efficiency of repair. And so we know that senolytics worked because, as compared to the old levels, which are shown here by the dashed line, after treating old mice with senolytics, many components of SARS, all of them that we tested, became diminished. So senolytics worked in this regard. But when we look at what happened to muscle repair, there is no improvement in muscle repair. So this is vehicle and this is senolytic. And if you look at the amount of fibrosis and inflammation here, it's the same. There are very few regenerated myofibers, as we can measure, and their numbers are the same based on the marker, embryonic myosin heavy chain. And this is all quantified data. So once again, senescence and senescent cells and senolytics tell one part of the story, and another interesting part of the story is independent of those. And so now shifting gears to fresh of the press, just in press, a story that has not yet come out on the online publication. I am going to tell you about our work on improving from predictions of chronological age to measurements of biological age. So that's a little bit shifting gears into the very last slide of my presentation today. And I will tell you first the team. So the work was done by our postdoctoral fellow, Dr. Da-Hwan Kim, who really did a remarkable job on performing all of the experiments and analyzing the data and putting that all together. And he was helped by Connor, and Connor is the PhD student who is interested in biophysics, but also is a brilliant mathematician. So he figured out how to analyze the data so we can measure biological age. And it is a collaboration with our long-term collaborator, Dr. Dobry Kiprov, and director of the UCSF Brain Aging Center, Dr. Joel Kramer. So tell you a little bit, what is the difference between predictions and measurements? And what is the difference between predicting chronological age versus measuring biological age? I'm going to illustrate it with this cartoon of the, I guess, country fair, where some people, guessers, can predict many things about you, can predict your height and your weight and your age. So let's say that I wanted to predict person's weight. And I could do it using population statistics methods, for example, by looking at their body shape, also by looking at their clothes, and perhaps knowing what they're eating. And believe it or not, if I knew many, many parameters and had many people for my statistical analysis, the prediction would be rather accurate. But it would never be as accurate as if I had the scale and I just basically measured people weight. It would be much more accurate. And furthermore, it could be done in a blinded analysis. I don't even have to look at the person. I can just look at my scale. So for many reasons, it's more important to measure than to predict. And we had this opportunity to measure biological age directly from the studies that Day One did on the samples that Dobre provided us. So Dobre performed similar experiment in people, not experiment, similar studies in people. I wouldn't even say studies. So he performed the similar treatment or procedure in people that I showed you, which we call neutral blood exchange. In people, it's called therapeutic plasmapheresis, where old blood plasma or young blood plasma or middle-aged blood plasma in people is diluted with saline and albumin, and the cells are also returned back to them. So Dobre performs this procedure routinely in his clinic, and he generously provided us blood samples from participants or from patients. We didn't know anything about the patients except that their age and their gender. And then Day One performed the studies where you can see visually, and this is proteomics analysis of blood proteome, that old people start here, and then round 0 or people before their plasmapheresis procedure are next. And then there's rounds of plasmapheresis, round 1, round 2, 3, 4, 5. And then young people are at the top. And what is very, very clear visually, you can just see it, that aging in this red and orange squares is gradually diluted. And though people are chronologically the same age or similar age at around 0, they become biologically very similar to young individuals. So it goes from these colors to blue. So we are diluting aging and we're making people younger biologically. And this is very, very clear, right? So then, of course, we ask the question, can we measure it directly? Instead of predicting what happens, we have all of the experimental data here. Could that be an experimental hypothesis that allows us to measure this dilution of old age? And then Connor had this brilliant idea of how to filter out this proteomics data on thousands of proteins and find the biomarkers which allow us to measure biological age. And so his idea was very good and very accurate. And here is the direct measurement of biological age of people, which does not rely on machine learning, data processing, or adjustment, and it is not a prediction. And furthermore, it is not a prediction of chronological age, but direct measurement of biological age. So each dot here is different individual, and what is particularly interesting to me and kind of reassuring to me is that it is not perfect. So our R square is only 0.72 because it should be like that for biological age. We don't have chronological age as main variable, it's biological age measurement. And young people are of a similar biological age. And as people become older, now there is big variability. Some people are actually younger and others are older. And you all know that, right? There are some people who say, oh, I don't believe you are 40. You look like you're only 30. And other people, oh, you look much older than your age. You need to take a rest. And the same variability increases even further. So the line is not perfect because we don't expect it not to be perfect from what we know about aging, biological aging. Then we analyzed the samples from dual Kramer group of people who have mild cognitive impairment. MCI stands for mild cognitive impairment on the same line as shown here. So this is the progression of biological age in control group. And as you see, people with mild cognitive impairment, some of them are much, much older. But what is another cool point is that some of them are in relative terms are 250 years of age. And of course, they are not 250 years of age everywhere. And they're not going just to turn into dust like in sci-fi movies when the spell is broken. But what it means is that for few parameters that we measured, in a relative term, their biological age is that old. And because for those few parameters that are in bloodstream, their biological age is that high, it means that overall, their tissues and their organs and organism is less healthy and older, not by 250 years. And then in the next experiment, we now compared the samples before therapeutic plasmapheresis that Dobry gave us, and after the very last round of therapeutic plasmapheresis, longitudinally in the same individuals that Dobry shared with us. And what you can see here, this is mirror images, right? This dot is one individual. You can see, you can see that the age has been significantly reduced by plasmapheresis in every single individual who underwent this procedure. Once again, if you look at the biological age, it's relative. It doesn't mean that this person is 15 years old and this person is 10. What it means is that in relative terms for those parameters, that is the biological age. So we cannot really think about biological age in the same way as chronological age. It might be very different. By the way, we have seen it before in other experiments that when old cells or old tissues in animals are exposed to rejuvenative treatments, such as neutral blood exchange, for example, the outcome for many proteins and genes is not that they are simply returned to the young state. They are super young. They are younger than young. But remember, this is not for the entire body, it's just for those few parameters. And once those few parameters are younger than young, it means that the entire tissue perhaps is just a notch healthier. Something for us to consider as a new paradigm of when we actually measure biological age without predictions. And so then how about all of these things? So we have also mitochondria and other organelles, metabolome, very complex, cytoskeleton membranes, proteostasis, extracellular matrix, and so forth. What we believe in our group is that dilution of old blood and perhaps then more precise methods on specific attenuation of particular factors is the skeleton key that will reset all other small things and cells and tissues back to youth and health. Of course, that has to be tested. And so here are my conclusions. First of all, large dilution of systemic milieu stably attenuates age-elevated inhibitors. Remember that there is no cell or protein or gene that evolved to make us old or sick. So we cannot simply get rid of them. At young levels, they are vital, but they are excessive, and attenuation of them should make most or perhaps all organ systems rapidly and robustly younger. Old blood dominates over young in vitro. That's very important to know. Blood dilution does not act simply by attenuating SAS or senescent cells. And senescence is not entirely cell autonomous and nor entirely time damage dependent. Senescence instead can be induced in 2 weeks in young mice with their young blood, young cells, good telomeres. So that again poses a question, would young blood help to old individuals when even young mice, which have still 50% of their young blood and all young cells and tissues, become aged just from one transient exposure to old blood? So another point is that we know our chronological age, and there are many very, very good machine learning models of how to link chronological age to perhaps additional biomarkers. For example, Steve Horvath, I'm sure, gave excellent keynote presentation yesterday. But might it be more meaningful to start directly measuring biological age if we can? So I would like to shout out many thanks to our laboratory, and this is before pandemic. This is, of course, during pandemic, diehard people who work in the laboratory then, and incredible research group. I'm truly blessed having them. Many thanks to our funding sources.